I'm going to introduce you to the next speaker. And I was just joking with you on Dr. Pascal's work. I won't keep you in suspense. I would let him talk about it. But I told him that probably you're trying to get me out of the transplant business because you're probably going to have nobody dying of brain death anymore. So please welcome, I would love to welcome Dr. Pascal here and speak about his work.
Well, thank you so very much, Dr. Bonde, for inviting me, and thanks to all of you for attending. I think that there are two reductionist pathways in biology. One is chemistry, one is physics. That's the way I see it. And I spent my whole career going through the chemistry pathway, and recently I became convinced that physics, meaning something you can manipulate or handle or modify in some way, it's probably more meaningful to the patients in the short run, because chemistry, as we know, it's extremely complicated and wobbly and redundant and full of surprises. So I've become convinced that engineering, it really is the way to go. So what I'm trying to say is, to me, represents a very new field. It's something that we didn't have a lot of experience with, so I apologize for any mistakes that I'll be making. I'd like to begin from the usual end, which is acknowledging people who did the work. A number of very talented, excellent engineering students. Now they've moved on to other things. They continue to be associated with the laboratory. And then a number of people who have been doing a lot of the actual work in terms of the surgery, and without whom none of this could have ever been possible. We work as a large team, not least of which is our machine shop person, Jimmy. There on the right, he's great at creating prototypes of things, and then my colleagues, Joe and Stuart, are great at fabricating microelectrodes, and it'll become clear in a moment why they're so fundamental. I also want to acknowledge my mentor, Buzz Brown, who was in fact a cardiac electrophysiologist. Some of you may know him, excellent researcher, and I'll be grateful to him forever for taking me in his laboratory for my PhD when he never took PhD students. So I will be immensely grateful to him. I have nothing to disclose. I actually have a lot of things to disclose. I just don't want to tell you about what I want to have to disclose. And then a couple of publications where you can pretty much find everything summarized, everything I'm going to be presenting. And I'll be presenting a few more things in addition to these two items. I'll be talking about why is human brain activity similar to pig brain activity. That's been a pending question in the field. Is a pig a significant model in terms of brain function? Is it or isn't it? And why so? And second, can the pig brain be maintained in isolation from the rest of the organism for experimental purposes? So that's the essence of the presentation. And so it occurred to me that there are three main questions in conventional bypass. The first question is this long-standing debate of pulsatility relative to laminar or steady flow. The second question is perfusing according to individual values that a person may have, as opposed to generic standard values that we may impose onto that person or machine or moment, meaning personalizing to whatever the flow or the perfusion happens to be for that person right there, right then, as opposed to arbitrarily picking a value. And the third question, which is very important to me, it's the neurophysiological activity. Obviously, we're talking about the brain. Does it change? Does it not change? Is it relevant at all? And so the students gave me this slide in terms of discussing conventional bypass. The numbers look very high to me. I hope they're true. They claim this is all published data. At my institution, this doesn't happen. I hope it doesn't happen at any other institution, but I believe this is published evidence that there's a significant chance of a mishap with a conventional bypass. So we're really shocked by looking at these numbers. So again, hopefully this is not totally accurate. If it is, it would be a significant problem. Now the issue is there is too many biological factors involved, too much chemistry, in my humble opinion. And there are many studies, and there are many cell components and processes and molecules, to the point that you start finding contradictory evidence depending on the cell type, the animal model, what you're looking for, It's just not easy to make sense of a lot of the data that's out there. If you look at the very basic biology or biochemistry or signaling or phenomenology in terms of the very low-level biology. And so it occurred to me that the way to solve these seemingly unsolvable conflicts, when you have different opinions or different approaches and they somewhat conflict. I mean, we all face this every day in medicine and science. There will be studies talking about something and the next study will differ to some degree. It occurred to me that instead of looking at all the studies and picking the better one based on whatever we might feel it more solid or more robust than the next study, I don't think that's where the solution lies. The issue is not so much how are they different from one another. The issue is what do they have in common and then reject it, because that's where the problem is. And so I don't think that needs any explanation. The second one is from my favorite bookstore in England, but I think that explains itself. And so why am I talking about brain activity? We took advantage of persons who were going to receive neurophysiological monitoring for days, sometimes for weeks, to identify an epileptic source within the brain. So the cranium will be open. They will have electrodes on the surface of the brain. These are these square grids with multiple dots, which are the recording electrodes. The yellow one happens to be one particular point that I'll show you in a moment in comparison to the pig. And because this happens over days or weeks, the activity in these people is awake and presumably normal activity. These are failed monitoring studies, meaning they did not have a seizure. All of the neurophysiology looked normal, which wasn't the intention. The intention was to characterize abnormal activity, seizure focus, so that they could proceed to surgery. But a fraction of these individuals have a failed study because they're quite normal throughout, and that's the data we took. So normal-looking human brain activity obtained in the awake state for a number of days. And then you can do that also in the pig. There's a way to proceed and do the same type of implant in the animal. Of course, for that, we have to create things like a Faraday cage you see there open and develop a number of things just so we could do a similar thing to the animal. That's my colleague, Bruce Mickey, implanting some of the electrodes. And that's what the pig looks like, not quite like the people you saw, but on the left, is a top to bottom X-ray where you see a few spots which happen to be the electrodes. Look at the yellow one; it's one of them. So that's the same one, well, equivalent to the yellow one you saw in the 4 persons' heads. And then you see a number of them that penetrate throughout the depth of the brain. Some of them are on the surface. So it's a fairly comprehensive. I think you can. I don't know that you can see the mouse here, but it's a fairly comprehensive. The penetration of different levels, so we can record different kinds of activity. Everything with an arrow, it's pointing at a recording shank or shaft array going into regions of interest within the brain. And then, of course, we can also utilize very different electrodes that we believe cause minimal damage to brain cells through the process of inserting the electrode. There's a long-standing problem in the field that many of you do know, which is that the moment you implant a brain with a recording electrode, because of the width of the electrode, we're talking microns in terms of brain tissue structure, there's significant disruption. So a lot of the data that you will read about is not really so much data about brain activity, neuronal activity. It's actually data about microhemorrhage activity when you're implanting a relatively sizable probe into a complicated structure. So what my colleagues did was to fabricate probes which are made of amorphous silicon carbide. And they happen to be, you can see on the bottom left, it's a 10 micron scale, and on the right you can see 3 images of I think we did all of this in mouse, just as an example with a 50 micron ruler, so you can have a sense about the size of an individual neuron in relationship to the size of the silicon carbide probe on the left. And you can infer that when this thing penetrates the brain tissue, cells actually do not. We've proven this, and they do not get damaged. There's no microhemorrhage. Cells literally just move apart, and the electrode penetrates. And so we believe we have a superior quality of a single cell recording. So again, we're going to be looking at single neuron activity within the brain at multiple levels, and hopefully without too much in the way of damage to those cells or anything else. Ideally, that's what we were looking for now. So that's a very complicated slide, but on the bottom left, you see a typical tracing of this sort of brain activity. This happens to be surface and depth, global activity. These are not single cell data. This is more sort of large electrode data. And on the right, panel A, what I'm trying to show you there in light green, the thick line happens to be the average neurophysiological activity of a few human brains. The thick line is the mean. The two thin lines above and below happen to be the standard deviation. And then in blue, you have the pig activity of the cerebral cortex. So they're very much superimposable. Now, does anybody know why the human brain activity has a peak at about 8 hertz? So we divide activity into frequency because it oscillates, and we divide the brain wave into different components. At 8 hertz in the human data, the green little peak on A, there's a little surge of activity. Does anybody know why people have that and the pig doesn't show that? The eyes are open. These are awake people who are looking around. The eyes of the pigs are closed. So you lose that activity that comes from the occipital lobe from looking around. And so years ago, we got very interested into looking at what particular cell type within the brain happens to be more vulnerable to metabolic failure. Metabolic failure meaning energy metabolic failure. And we proved in some models and also in people that inhibitory brain cells happen to be the first to be injured or damaged or fail or deficient whenever we depress energy metabolism. These are genetic models. These are not experimental models in any other way. But the point was made, or we made, that inhibitory cells happen to be more dependent upon energy metabolism, including blood flow, than excitatory cells. So they happen to be much more selectively altered. And that's important because in brain circuits, inhibitory cells keep excitation in check. If inhibition is suppressed, as you can imagine, the balance from excitation to inhibition shifts, and then excitation will run off, will run away. And we proved that happened to be related to a particular synapse that's really very sensitive to metabolic failure. And the point is not so much to highlight that, which is well known by now, but to mention that if you manipulate the human brain, or the pig brain for that matter, during one of these procedures, we want to be looking at inhibitory cell activity in addition to every other cell activity, but we're going to pay attention to inhibitory cell activity because we know it's highly vulnerable to ischemia and to manipulation as a readout of brain integrity, neuron integrity. And so it's obviously possible to do this in the animal. We just have to occlude a blood vessel. That happens to be the medial cerebral. Here on the arrow, it's that animal happens to have one medial cerebral. Often they have two, sometimes they have three. I think we've done this 35 times. So one pig had 4 medial cerebrals. They're very different from us, but whenever we find the vessel, we go ahead and clamp it, as you can see here. And then we leave a clip for a period of time, and then when we're done, we release a clip. So we can occlude the blood vessel, pretty much causing ischemia in that brain region, and then when we're done, we remove the occlusion and see what happens. That's what we call reperfusion, which is so well known to all of you. And so what happens, just to make it brief, is that there are very significant, very quick changes in global brain activity the moment we occlude such a blood vessel. I'm just trying to validate the model. I'm just trying to argue that the model is quite sensitive to ischemia along the lines of global brain activity as measured by these electrodes. So you can see the clamping happens at some point, then we reperfuse, and then other components of the brain activity begin to change as well. So it's a very sensitive model to be looking at perturbation of blood flow to the brain. I should also say, the moment you clamp the medial cerebral in the animal, we have extensive neurophysiological changes across the whole brain, which is unaffected within seconds. which was a major surprise to us. So not only you have this function where you will have the injury or the ischemia or the stroke, but you also have global brain changes, and they actually evolve over time, which is a sort of interesting to us. Brain is a network. And now we also couple that, and I'll be talking about this very, very briefly, we also couple that with taking microbiopsies of the brain after infusion of carbon-13 molecules, because we can do NMR on the brain tissue and be able to identify carbon flux from glucose to lactate to glutamate to GABA and so on, to the many different molecules that we're interested in. Not only abundance of these things, which are very important, but also flux. At what rate does lactate begin to rise, and how does it do so, and from which molecules? That can only be done by NMR or mass spectrometry, and the data looks like that. This is not important. The important point is that on the right, even before we see an increase in lactate production, we already see a decrease in GABA, which is inhibitory cell neurotransmitter production. So even before lactate goes up, this inhibitory cell neurotransmitter starts to be suppressed. I'll go through this very quickly. We can talk about it much later in great length. And then finally, we have, this is a very difficult slide also, 3 panels on the left. These happen to be single neuron action potentials taking place in the pig brain. In A, left column, before we cause the occlusion, middle column during the occlusion, right column, the occlusion. Every line will be a mean and a standard deviation, and on the right, these lines happen to be action potentials, so neurons firing at some frequency. Some of them fire all the time, some of them fire and then they pause. They come with a firing frequency, they come with a shape of the action potential. These are very well known, well, they're not very well known. Now they're well known parameters that we look into because we're interested in the ideally the preservation of this single neuron activity firing, which reflects synaptic activity. So if that is preserved, if that continues to be the case when we do what I'll show you in a moment, then a lot of things are functioning the way we hope they will be functioning. At the bottom, you see the electrodes have been implanted. They're actually invisible to the eye because, you know, at 20 micron electrode, it's just not visible. So it's a challenge to be able to insert it into the brain. And then we also finally couple that with single-cell mRNA analysis, because we already know from other kinds of studies from many other people that the moment you have ischemia or hyperperfusion or injury, a variety of RNAs begin to change within neurons, and this is a representation of the whole brain mRNA landscape, and we can sort different cell types based on their mRNA content. Again, not very important. What I'm trying to say is that we can identify inhibitory cell mRNA changes very quickly the moment we have ischemia. And so we have another method yet to look at the preservation of brain activity. So in conclusion from all of this, I can tell you confidently and briefly that pig and human brain activity are not distinguishable for our purposes. And second, I can also tell you that these sort of activity, these sort of phenomena that I showed you are very sensitive to ischemia. That will not be new to many of you who in the 70s had read the endarterectomy literature, where you clamp and within seconds you have delta EEG changes in the electroencephalogram. That's been very well known for decades. But here is some of the underlying mechanism for why that is the case. And here is how we can even refine that approach to prove that we can do this. And this is just to replicate what's been done with individuals in the pig. And again, the goal eventually, or continues to be, to isolate the pig brain into a machine for research, for reasons that I can go into if you have questions. But the goal was to be able to isolate the organ in a way that we can perfuse that will be near natural using an artificial machine to keep blood flow to the brain, and all of these activities should ideally be preserved throughout. That's our objective. And to do that, obviously the circuit and the mechanism is not trivial, as you all will know, but it has pretty much the same components of a bypass machine with a few more control mechanisms. And the whole summary and the whole difference about this whole thing is that we adapt the flow and the pressures we measure in each individual close to the brain to the output of the machine. So we measure what's happening here or here, doesn't matter. And then the goal is to have the machine individualize the output of that circulation, just so we can preserve what the animal had right here before we began the surgery. Is that clear so far? So it's individualized to the specific animal, and we find a significant variability from animal to animal. They don't have the same blood pressure. They don't, the things vary. I presume persons who are going into a bypass machine are not likely to have the same hemodynamics here in their neck. Probably not. Perfusion of the brain tissue, we think, ought to be constant for everybody, but how that perfusion gets there through the vasculature may be very different. And so we wanted to individualize to each animal or each individual, as opposed to applying a generic flow process. And of course, it comes with a significant control machinery, just so we can feed back and feed forward into the appropriate parameters. So the way it works, we measure, as I said, the native waveform. In this case, I think this is pressure, and then we're looking at typical pressure reading. We isolate one waveform that we may like, a representative one, looks that way, and then we go ahead and convert that into voltage, idealize that waveform, and have the machine replicate that output to what the animal is. This is what the machine looks like. It needs to be made smaller, needs to be made look nicer. And I'll show you one by one the different stages, and I'll go again quickly through that, and then we can go for questions. So we do anesthesia. We try to be very mindful of not interfering with neurophysiology because of the anesthesia we're utilizing. So obviously, it's important to not suppress brain activity, because, as I said, this is pretty fine-grained, detailed neurophysiological activity, which is very prone to be disrupted just by anesthesia itself. So we're mindful of using anesthesia. It took actually years to come up with an anesthesia regimen that would not interfere too much with the neurophysiology. So this is all sequential steps that are taken, and then what we do is we go ahead and initially implant the appropriate electrodes in the cranium. Brain will be exposed. Oftentimes we end up decapitating, and you have a base of the cranium and some of the vasculature, but the brain will be, at that point outside the body. And then we have a couple of approaches. One approach has been to perfuse into the aorta, and another approach has been to perfuse into the brachiocephalic. And of course, these are just entry points, which are then ligated proximally and distally appropriately, just so we can devitalize the body and just leave behind circulation through the carotid. So in there, I'm showing you some of the data that's acquired from the system. Some of that is flow, some of that maybe pressure, whatnot. And then I'm showing you the bypassed access and egress points that we have chosen. I'll go through that in a bit more detail. And of course, that's coupled to the neurophysiology that's ongoing in the meantime. So here's the approach of stepwise. We also mind things like brain temperature, pressure very much. We mind many other things that come with having a brain that's going to become very dependent on the machine within a few minutes. And we like to keep track of all of these parameters so that we know what we're doing with the perfusion. So in terms of how this works, this is one example of perfusing the aorta. It's self-explanatory. You approach the common carotid to be your readout. So we're going to be isolating or measuring that flow here before we do anything, and that's going to be our end goal, which will be to replicate that flow, that pressure right here when we go on the machine. So we have that data for that individual before we go ahead. And then there'll be obviously surgery done, things are ligated, so at that moment the body stops to be perfused, and then it goes into the machine. So what that looks like, some data for those of you who are very data-minded. There's a number of values that we measure from the animal. We select a particular segment of these values, and then we go ahead under EPCC. That's what the machine is outputting into that perfusion mechanism. So the fidelity is fairly, reasonably accurate for our purposes. Again, on the upper left, that's a waveform that we have for pressure in the aorta. On the right in pink, on the top, that's a waveform we have for pressure in the carotid. On the bottom, you have a single waveform. The black one, it's the native. The blue one is what the machine is delivering at that level. The aortic waveform doesn't really quite match the native waveform, but if you look on the right, which is what happens in the carotid, the matching is much closer. So what I'm trying to say, to have a carotid pressure cycle that's very similar to the native pressure cycle an hour before the surgery proceeded, that's what you need to be inputting into the aorta on the left. So the waveform of the aorta doesn't need to be identical. First of all, because you have a different aorta now, it's a fragment of what it used to be, so the waveform that would not have to be similar to that one. But that guarantees you have this carotid oscillatory pressure waveform, which is very close to what the animal had before the surgery. In the second example, here's a brachiocephalic approach. The same thing, flow, pressure, things are determined. They're put into the circuit, and then the circuit will output the parameters with the pump, retaining this sort of pulsatility. So the same thing, brachiocephalic pressure on the upper left before surgery, carotid on the upper right before surgery, and then after that, below, the circuit is able to deliver in blue that sort of waveform, and on the right, that waveform at the carotid. So the fidelity of the perfusion, we feel it's adequate for what we're trying to accomplish. And then the important thing is, did the brain suffer as a result of this? Is any of the aspects that I introduced you to a moment ago, and any of the array of things that we measure, change in the brain? Because our goal, again, our hope is that throughout all of this, ideally, the brain should not know that any of this happened. I think that's the ideal world. And so we go ahead and apply all of these after the fact, just to see whether we have any repercussions on brain function, broadly defined according to the many criteria that I told you about a moment ago. So what we do, that's also, significant, hefty amount of data analysis that has to go into this, because the different components of brain activity arise from different cells and relationships between cells. So it's not totally clear, and there are oscillatory waves in the electroencephalogram, the electrocardiogram. It's not totally clear the oscillatory waves, where they come from, the cell types and the relationships. It's not solvable, but we want to deconvolute the electrocardiography, or any neurophysiological signal for that matter. We want to deconvolute that signal, which will be oscillatory in nature, into subcomponents, so that we can then go in and look into each of the subcomponents and see whether any of them will be affected. Why? Because the subcomponent that we have proven to be related to inhibitory cell activity happens to be a subcomponent that oscillates at 40 hertz, 40 to 45 hertz. So inhibitory cells fire very frequently. They fire 100 times per second. They're very metabolically active. As I said, they're very vulnerable to metabolic failure, ischemia. Likely that's why they fail before any other cell type, because they're so metabolically needy, because the discharge rate is so large. I mean, 100 times per second is a very significant firing rate. Imagine the gradient of sodium potassium that has to be replenished every couple of milliseconds to be able to maintain that action potential rate. And so that inhibitory cell vulnerable activity happens to be reflected, we have shown, in the electroencephalogram and in the other activity as a 40 hertz oscillation that we can detect even with a surface electrode. So we want to look at the 40 hertz component of the electroencephalogram, which is important for that reason, but the 40 hertz, it's hard to measure. there's interference from 50 hertz noise. You need a Faraday cage. It's an infrequent event. So essentially it takes quite a bit of effort to deconvolute the EEG into the different kinds of waveforms that we like, just so we can talk about inhibitory cell sources of the electroencephalogram. And that's represented in here. The mathematical treatment, it's been also published, and we've been working on this very heavily through a different separate body of work. And what happened is, just to make it brief, on the upper left for subject 1, left panels, and for subject 2, right panels. On the upper left, you have the, I think this happens to be depth electrodes in the hippocampus. So for subject 1 on the left in panel A, and for subject 2 on the right in panel A, you see the native electrical activity within that region. You see before EPCC, and then you see below after EPCC, and on the right there's a comparison. They're really indistinguishable in terms of activity. So across the whole range of neurophysiologic activity for that pig brain, for that particular animal that we put into the machine, the activities that we measure at that level, which is the hippocampus, which happens to be very vulnerable to ischemia. Every physician here knows very well that hippocampal injury is a leading cause of disability after ischemia. It's essentially unchanged once we go on the artificial extracorporeal pulsatile circulatory control. Below, in colorful part of the chart, shows you the different subdivisions of the brain wave into these delta, theta, alpha, beta, gamma components to show that they are essentially unchanged before we are on the machine and after we go on the machine. If you pay a lot of attention on the left panel, on the bottom, you see the pink gamma activity. After a while, it goes up and it becomes flatline. Somebody flipped a switch. This is so sensitive to people walking with high heels or an elevator moving. It is difficult to isolate these signals because they're low in amplitude, very high frequency, as I said, plenty of interference. So, but we never modify the data. We never, by the way, we never pick data to show representative example. We just randomly, intentionally pick any segment of the data at random to analyze it and to publish it. So we haven't selected for anything. Somebody flipped a switch in their own time here. You will see that in the analysis. So subject 1, again, it's the aortic axis. Subject 2 happens to be the brachiocephalic. And we're quite comfortable that both of them didn't see much of a change when we went into this device or perfusion method. So what do we need to do next, or what do we want to do next? So we want to, obviously, now what do we have? So we have a brain that so far we have been able to maintain for, I think we've done 6 hours or 7 hours at a time. We haven't gone beyond 6-7 hours. I presume we'll run into other difficulties if we go beyond a 7-hour window, of course, with perfusate and all kinds of things will come into consideration. But now we have a brain that's essentially complete access because we have removed most of the cranium. So now we can access the brain to do neurophysiology. We can do metabolism that I showed you a moment ago with neurochemistry. We can do drug studies because now obviously there is no body anymore, so we don't have to, we can ignore hepatic metabolism, renal activity and so on, because you have an organ that's going to be essentially isolated to some degree. And then we can look at the consequences of, for example, a stroke in an organ that's separated from the rest of the body, which I think has really been a challenge for us who are clinical neurologists to be able to identify what's really, what are the symptoms that you can attribute to the injury in the brain relative to everything else within the organism that changes anytime you have a significant brain problem or a brain disease. So that's been a long-standing source of problems for us when we try to diagnose stroke and many other diseases. What's really brain-related and what's really everything else impacting the brain in ways that we don't fully comprehend. So I think having an isolated or nearly isolated preparation will help us get into this. If you're interested in, studies from the 80s, for example, it's well known, and I'll close on that note, and then I'll take questions. It became clear in the early 1980s, for example, just to give you an example, two examples. It became clear even before the 80s that individuals who have profound hypoglycemia, human beings, so I'm talking about zero blood glucose, I'm not talking about mine, I'm talking about zero blood glucose. Who are they? Well, insulin shock people, failed suicide, alcoholics. These are people who will, on occasion, come to the emergency room in a coma with a blood sugar of 0. And you obtain an electroencephalogram in them to see if they're dead or alive. The electroencephalogram will be flatline. Flatline as in brain death criteria. So, high amplitude of the waveform to be able to see any activity and there's no activity. You can do that for quite a while. And if you apply glucose, administer glucose after that, the person fully recovers. So you can have zero activity and then the person fully recovers after that. There are alcoholics who, in our institution, we know them because they checked in periodically in a coma and blood sugar 0. And they come in, they put them in there, give them glucose, wait half a day, then they leave and they come back the following week again. And they don't eat. And people who overdose on insulin. So the brain will tolerate some degree of hypoglycemia or aglycemia, provided you go ahead and rectify that. And that's become known in the rat in 1984 in terms of how long will the brain take with zero glucose applied. Well, all of those studies have to do both with brain activity, brain glucose, of course, but they also have to do with every other change that happened in the organism as a result of aglycemia, of course. There are many things that are happening. There's ketone body production, there's lactate production, there's a suppression of insulin. What I'm trying to say to you, this sort of physiology cannot be approached in a reductionist way, in a scientifically controllable way, unless you isolate the organ in question, like you beautifully talked about a moment ago. You have to isolate the organ in question so you can decrease all of the other influences, because the organism is, it's intended to be a symphony. It's not intended to be a solo. So on that note, I thank you very much, and I'll take any questions.